Good afternoon, and welcome to the Data Analysis 101 workshop, discussion on tools for network inference. My name is Tiffany Willoughby, Education Programs and Outreach Manager in the Office of Research Integrity and Outreach. I will be your host. Joining me as your presenters today are Dr. Faruck Morcos, Assistant Professor in Biological Sciences, Dr. Justin Ruths, Assistant Professor in Mechanical and Systems Engineering, and Dr. Tyler Summers, Assistant Professor in Mechanical Engineering. Doctors Morcos, Ruths, and Summers will individually share effective tools to use when analyzing data remotely. Our first presenter is Dr. Ruths. Dr. Justin Ruths is an assistant professor with appointments in Mechanical Engineering and Systems Engineering. He joined UTD in 2016 after spending 5 years as an assistant professor and part of the founding faculty at the Singapore University of Technology and Design. His research includes studying the fundamental properties of controlling networks, bilinear systems theory, security of cyber-physical control systems, and solving computational optimal control problems focused on neuroscience and quantum control applications. Dr. Ruths, welcome.
Thank you very much. So thanks for joining us, and I hope this is informative. I'll be kicking this off, but hopefully what you'll see is a couple of different ways that we're all looking at similar problem, namely a problem of inferring information about a network, about the structure. So the method that I'm going to be presenting today is actually not my own method, but it's one that I'm bringing into collaborations that I have at UTD. This one is actually recent work by my PhD thesis advisor, Jershin Lee. He's down here in blue. And so this is the paper. If you'd like to learn more and read more about this method, you can take a look at this paper. Shou Wang is a recent hire in Arlington, UT Arlington, and so she's actually nearby. But we found this method to be very useful and compelling option as we move towards applications that involve using data to infer networks so that we can use some of the other techniques that we've been developing in research. I'll show at the very end some ways that we're trying to extend this method to make it a little bit more useful. So it's worth just kind of spending a little bit of time thinking and defining what network inference is. So network inference, it goes by a number of different names. Sometimes people call it link prediction if it's done on a smaller scale. This is essentially the problem of going from some data into rebuilding or building from scratch a network that represents that data. And so again, this comes in many forms. One of the classical problems that maybe people are aware of is kind of the Netflix problem, which says, okay, I like this movie, predict a movie that I would also like, but I haven't seen yet. And so you can think about similarity as being a way of establishing connections. There, the data is about the type of movie, the actors and actresses that are in the movie, the content, who does the soundtrack, and so on. So there could be a lot of different factors, and different people like movies for different reasons, and so you would use that data to infer connectivities between networks. The types of data that I'll be addressing and I'm interested in are what's generically called time series data, and that kind of comes with it the idea that we have data from a streaming source or from a process that's exhibiting some sort of behavior, and we're recording that over time. And so that time series literally just means aggregated over time. And so one of the motivating examples that I have in the work that I'm interested in doing it comes from neuroscience. And so we have some collaborators in various departments at UTD that are interested in kind of bringing network inference methods to bear on neuroscience data. So one example of that could be electrocardiographic readings that you may get from inserted electrodes into the brain. It could be coming from EEG type devices, but the idea is that you have some sort of signal or signals that are coming in and recorded over time, and we're looking for essentially the network structure that gives rise to this behavior. and not only network structure, but also some of the dynamics. You can think about way back when, some of you maybe this is a long time ago, some of you that you deal with this every day, differential equations, for example, that represent the behavior of each individual piece of this system over time. And so we're fortunate to live in an age where networks are relatively well-established concepts. Networks are essentially nodes and edges, collections of nodes and edges. Nodes represent either people or agents. In brain networks, they could represent cells or groups of cells. And connections represent connectivity that drives the behavior, in this case, of the system. And so for anybody that's intimately aware, I just grabbed a picture of a network to demonstrate this. This happens to be a nematode network, so you really don't want this to be the representation of your brain if we were recreating your brain. Relatively simple example here, but also a well-known example from network science. So this is the problem that we're interested in. We're interested in establishing this network structure from this time series data. And so I'm going to start out with an example and kind of a proof of concept of a toy example to demonstrate how this works and how it works well, and essentially the process, and then we'll just dig into it. I'll try not to get into too many of the details, but I'm happy to go over it with anybody else that wants to follow up on this or would like to collaborate on this. I think that's largely the idea of this this series. So the toy example that I'll use is a collection of what's called oscillators. So in this case, they're Kuromotov oscillators, and that just means it has a specific functional form. What this means is that this functional form has that the time change of the state of this oscillator, it's described by a single number, theta, and that is driven based on some natural frequency as well as an interconnection term between other oscillators around it. And so people use this to represent all sorts of different phenomenon. One of them is neuroscience behavior, with the idea that an action potential of a neuron could be captured as a phase, starting from 0 and ending at 2 pi, and essentially repeating whenever that action potential happens again. So that phase, that cyclic behavior of the neuron, is captured with a phase. And so the evolution of these kinds of systems would look something like this, and this plot down below is the data that we'd be dealing with. And the idea is whether we can learn the connectivity, which is this K, which you can think about as being a matrix that defines how one node or one oscillator is connected to another, and also learning these natural frequencies that describe the natural behavior even when this oscillator is not connected to anything else. And so this is the data. So these oscillators start at different points. That's on the left-hand side. Time is on the horizontal axis, and these move along. And so this is the data that we want to fit in order to reconstruct how these different oscillators are connected together. And one of the interesting things as you work on this topic is that different segments of the time series give you different types of information. One of the characteristics of this type of behavior, in particular the difference, this theta j minus theta I, this is a diffusion type relationship between the oscillators, which means that they tend to reach a consensus of sorts. And so that means, as you can kind of see in steady state, as time goes on, they're all kind of moving together. They're not staying the same, but they're moving together. And so that actually gives you interesting information about the connectivity matrix, whereas the more jumbled activity that happens towards the beginning, what I'll call transient behavior, is really tells you something more about their natural behavior, this natural frequency. So there's different aspects of data that give you different information. So this is not actually the only data that we use. In fact, these problems are characterized by requiring tons and tons of data. So the actual data that I'm using, it looks more like this, where essentially I've run this simulation of this oscillator set over and over and over again. And so you can see it's rather dense, but we require this order of data in order to reconstruct all that connectivity. So again, I said I just kind of demonstrate how this works. So what we're able to do from that data, what we're able to reconstruct without any prior knowledge, except for that set of data, is the fact that the actual Kij matrix is in the center. That's our panel C here, and we're actually able to reconstruct that very accurately, and that's in panel B. And so this is what we infer. And what's also interesting is that we're able to infer the correct omega I, the correct frequency related to the actual frequency. And so that's in panel A. So one of the important things to understand and appreciate in the context of this problem is the fact that when you're dealing with time series data, it's very interesting or very compelling to think, oh, I'll just try to take a correlation between signals. So just because two signals are correlated, maybe that's a good indicator that one causes the other, for example. The challenge with that is that dynamics can be a lot more intricate than that. So in a very simple model, perhaps that could be the case. But oftentimes there are delays, there are nonlinearities, there's all sorts of effects that could actually invalidate this idea that we should just correlate signals. And so that's for example shown in panel D. Here we're trying to just use a correlation between the different theta values for the different, in this case, 32 oscillators that we're characterizing. And you can see that the correlation matrix here doesn't encode the same information that the actual dynamic connectivity matrix does. If we were to think about these oscillators as neurons and we look at their firing rates and we try to find correlations between their firing, then that pattern also does not give rise to the type of information that we're seeing in the dynamic connectivity. And so there's been a number of attempts at trying to use different ways of thinking about correlation, but in the end, it's difficult. It doesn't really capture the same kind of information that this dynamic information that you get from the actual connectivity does. And so that's our example. And so what I'd like to go ahead and do is just describe at a high level how this method works so that anyone that's interested could follow up on this. And so before we do that, what I want to kind of show you is and remind you maybe, or either again dig it up out of the past or something that's near and dear to your heart, is just the idea of the Fourier series. And so the method that I'll show you relies very much on the same ideas of orthogonality and projection that Fourier series does. And so at some point in your career, you may have come across the fact that you can represent a periodic signal very well using sines and cosines. And so the way that that's kind of formally written down is that if you have some sort of signal f of t, so this is a time varying signal, that can be expressed as a constant term plus a bunch of cosine and sine terms of different frequencies. So the different frequency is k. And the amount of those frequency is this ak variable and the bk variable. And those are what's called Fourier coefficients. And so these Fourier coefficients can be extracted essentially by looking at the projection, it's called, or the similarity between each cosine term and the signal itself, f of t, or the sine terms and that same f of t. And again, this really works because of the orthogonality of the functions that we're choosing. Importantly, not only sine and cosine are orthogonal, but different frequencies of sine and cosine are also orthogonal, mutually orthogonal. So it gives us the capability of expressing this signal, and not only doing that, but also doing that in a very compact way. So you need very few signals compared to if you use arbitrary functions to try to approximate this. So there's another way to interpret this, and that is that we can, and I know we're getting into a little bit of the weeds here, but it's worth just reflecting on this. Another way of thinking about finding those coefficients is to look at a sampled version of that function. So I'm going to sample this function at m, capital M, different points. That's t1, t2 through tm. And what I'm going to do is then I'm going to be able to reconstruct these coefficients, A0, A1, B1, and so on, by using a matrix that captures this sum that's in this first approximation. So this sum and all of these different sine and cosine terms can be aggregated in a matrix. And now this matrix, which includes all of these different frequency harmonic terms of sine and cosines, can these, the Fourier coefficients can be extracted from that and represented as this A matrix multiplied by a vector of these Fourier coefficients. And if we minimize the difference between our samples and our approximation is this AZ term, that is able to reconstruct those coefficients in a very, very similar way. And so that really is the basic idea. I'll point out a few very elegant things that the proposed method does to make that a lot easier for these network inference problems. So it's worth just mentioning that there are other orthogonal functions such as Legendre polynomials and Haar wavelets. And so when your signal is not necessarily periodic, instead of using sines and cosines, you can use these other function sets to represent functions compactly. All right. So getting more towards the method, the idea and the elegance of this method is that instead of representing just a function, we're actually representing the derivative of a function. So we're representing this, this time derivative x dot. So that's x is the state, and the dot means that we're taking that time derivative, and we're going to approximate this with an expansion, the same kind of functional expansion, where these qk's in the first term and the pk and pl terms, these are all functions that approximate the behavior. So I'm going to switch back to the Fourier example just real quick, and what I want you to appreciate is the fact that this is a function in time, and we're approximating it with functions also in time. One of the most elegant insights to this method is the fact that this is a derivative, which is a function of time, but rather than express it as an expansion of functions that have taken variables of time, you'll notice that qk, pk, and pl are functions of x itself. So we're representing the derivative of a state x, or xi in this case, which is a function of time, and we're not representing that as an expansion of functions of time. We're expanding that as functions of state. And so that's one of the elegant aspects of this work, and makes it actually robust to noise effects that may impinge on the system. And so essentially the way this works is that these bijkl, lots of indices here, unfortunately, but that's the way it works. These indices indicate, essentially iterate through the different terms that you have for the state, the different neighbor terms, as well as the different expansion terms. And so it just gets a little notationally complex, but in principle, the idea is the same thing, that we're going to find these coefficients ak and bijkl, and that becomes the part of the z vector that we're interested in expressing. And so let me just switch forward here. The optimization, just like before, the optimization that we're doing is this y minus AZ. The A matrix gets very complicated, and I've written it down in these slides, but it's not worth going into right now. I'm happy to share these slides with people if you email me. But the idea is that all these coefficients still reside in Z, and the A matrix still essentially compiles all of those orthogonal functions evaluated at your discretization points, at your sampling points. And so that the y takes on a slightly different value here. And the way that it works is that we take finite differences between our actual data. So our data is sampled at these time points. The orange is the actual curve behavior of the system. And so what we're doing is we're composing y out of finite differences between the actual data points. And that gives us an approximation of the derivative. And so now we're going to use that derivative in this optimization function to minimize the difference between those. So again, I don't want to go into the detail of all of the different terms, even though I put them on the slides. And again, I'm happy to share the slides with people. But that's the concept of the idea. And so what we're working on moving forward is integrating certain things like external effects, such as external inputs. So these are not taken into account in the framework that's currently there. Also, it assumes only single derivatives, so that precludes more sophisticated systems. And so right now we're working on, and we've kind of already generalized this for higher order systems, where we approximate with higher order finite differences. And so a van der Pol oscillator is a similar to a Kuromotov, but a more sophisticated. And here what I'm showing you in this image is that we're able to reconstruct relatively well. And finally, one of the biggest challenges that we face in real-world network inference problems is the notion of sparse measurements. Right now, we're assuming that we can get measurements from every node that matters. And so dealing with that sparsity is actually another way of thinking about even higher order systems, but that's a big challenge moving forward. So that's a brief recap of this method. Again, I can go back here to the original paper so you can get that reference down, and I'm happy to share these slides with anybody or have extended conversations with anybody after this series is done. Thank you.
Thank you, Dr. Ruths. The first question is, what is a Kuromotov oscillator named for?
There's a person, that's a person's name. I forget his first name, but this has been around for a little while, and so that Kuromotov is the person's name.
Excellent. The second question, for the Fourier series, why do you need as few signals as possible? Does that make the data easier to analyze?
Usually you get computational gains and you get more explainable phenomenon if you can reduce your signal down to the fewest number of terms possible. And so oftentimes representing an arbitrary signal as compactly as possible is very interesting. And so that's the justification for using what are called spectral expansions, where the elements are orthogonal to each other. That gives you that kind of computational, as well as I think kind of easier way to explain a phenomenon if you just bring it down to its most fundamental parts.
Thank you, Dr. Ruths. We appreciate it. It doesn't look like there are any additional questions at this time.
Well, I'm happy to have conversations after this or at the very end of all the presentations.
Very good. Thank you very much, Dr. Ruths.
Dr. Faruck Morcos is an assistant professor in the Department of Biological Sciences, Department of Bioengineering affiliate, and member of the Center for Systems Biology at the University of Texas at Dallas. He joined UTD in the fall of 2015 after postdoctoral work at the University of California, San Diego, and Rice University. His research interests include solving problems at the interface between biology, computation, mathematics, and biophysics. Currently, his lab focuses in developing methods to extract biological information from sequence and genomic data and create models of molecular evolution, protein structure and function, as well as bimolecular interactions. Dr. Morcos, welcome.
Thank you, Tiffany. Thanks for the introduction. I'm glad to be here. I'm gonna share with you some slides that are connected to what Justin was mentioning. So my talk is going to be more specific. So Justin mentioned a very generic approach to infer connectivity. In my case, I'm going to share with you some more specific applications, but from kind of a conceptual point of view, they're very interconnected because the idea is to find connectivity among nodes in a network. So for the case of biology, it's been over and over proven that networks of interactions are really much more important than just the sum of its parts. So basically, this complexity is extremely important in biology, and understanding it is one of the main pursuits of modern biomedical sciences. If we only have the individual nodes in the network, then we miss a lot, so we don't have a complete picture of how living systems work. And having at least a better understanding of the connectivity is going to help us understand biology in a better way. In our lab, we care about molecular networks. So biological networks could be very diverse. They could be at the scale of communities or ecologies. They could be at the scale even of societies, or they could be at the scale of atoms, right? Atoms interacting. And in our lab, we care about molecular networks. So these are interactions happening among molecules, or at least subunits of molecules that are atoms. And we want to build better methodologies, or at least methodologies that will help us analyze current existent biological data. We also want to create tools. So one idea is methodologies, but sometimes these methodologies are hard to kind of implement, right? They're hard to use. So we also try to make an effort to create tools that are easy to use for other scientists, especially if they don't have the background needed to kind of build it themselves. So we study those networks at kind of two different levels, two different hierarchies. One is connected to how the biomolecules, in this case, let's say proteins, how their building blocks interact. To give you an example, an amino acid, a protein is a chain of amino acids, and we care about how these building blocks, these amino acids, interact. So finding physical interactions or functional interactions is important, and we care about kind of revealing this type of networks. Then we can also go to a little bit larger or higher hierarchy, where we care about how these biomolecules interact. Not their building blocks, but how the whole molecule, like a protein, interacts with another protein. This has been shown to be extremely important for biology, because regulation of gene expression is all about interactions, and the building blocks of the cell are built through complexes. There's also the idea to understand how proteins interact with RNA and other molecules. There's also the idea of communication. So cells transfer signals, and then this kind of communication system works through physical interactions, or at least through networks. Okay, so today I'm going to discuss a couple of tools and methodologies we have been working for some time, and some that are new related to connectivity. One of them is called direct coupling analysis, whose aim is, in a very analogous way as the paper that Justin described, its aim is to find direct correlations rather than any type of correlations. We want to have direct connectivities or couplings among the input data that we have, and in this case it's mostly biomolecules, sequences of biomolecules. And then we have another tool that has to do with this second hierarchy of how biomolecules interact, and this deals with a very specific problem of signaling networks. So one type of protein called histone kinase interacts with another protein called response regulator, and we're trying to make a tool available so that people interested in this system can make inferences on their links, right? This tool is called Elixir, that's how we call it. Okay, so let me start by talking about direct coupling analysis. This methodology has been there for a long time. So I helped develop this methodology, and there's others in the field that are also contributing. But the main idea is to find direct correlations between amino acids in sequences. So I'm talking about availability of data. So if we have a collection of many sequences that encode for similar proteins, how do we find what are the important connectivities among these amino acids that will represent something connected to function, right? Now, these direct couplings turn out to be pretty informative of many structural biology questions. For instance, if we know how this connectivity matrix, as Justin described, if we know how this connectivity matrix among the positions in the sequence is distributed, then we can say something about how this protein is forming a 3D structure. So if you notice here, here in in a video that I'm showing here, this is the structure of a protein that's shown in blue, and the red dots, sorry, the red links are representing the top most important connectivities among those positions. And if we take those red links into account, and then if we use them in a model, then we have shown some time ago, and even right now, some folks at Google have used also this idea to predict protein structures, right? Because you know what are the important connectivities and how they should be close by in the tree structure. But we also know that this same idea can be applied to complexes. So here I'm also showing a picture of two proteins and that form complexes, and basically by linking their sequences and then understanding this from the point of view of their direct connectivities, then we can find interfaces, how these proteins really interact in physical space. The background, the mathematical background and computational background comes from the information theory and also from statistical mechanics, but ultimately is very, very connected to the problems that Justin discusses. Can we infer some connectivities based on the data? In this case, rather than having time series, we have sequences that have changed. They have evolved through time, but conceptually similar. So examples that can be studied with these methodologies, analyzing sequence data of proteins to find their known structures, as I've shown here, but also identifying interfaces that could be useful for drug design. To give an example, right now everyone is interested in COVID-19 and how its spike protein interacts with the ACE protein in humans. And this is one example where knowing the interface between these two proteins will be very relevant, because then you can design drugs that could kind of get in the way. Now, this is a tool that is already available for the scientific community in both as a downloadable code, or there's also, we're collaborating with the Rice University. We have a web server called dca.rice.edu where people can go and then get the analysis done for them in a web server fashion. Now, because this data, the output of this methodology is basically raw data, we have also developed with Warsaw University a plugin for a very popular tool in molecular biology called Pymol. It's a tool to visualize proteins, but we have added a plugin where you can take this output, this connectivity matrix, and then compare it with the tree structure of a protein and one-to-one. So in this case, I'm showing on the left is an example of a matrix. The lower triangular part is an actual crystal structure. So these are the X and Y axis are residues in the protein. The dots in black are residues are in physical close physical space in the crystal, in the tree structure. And then on the upper triangular side, we have the output of this or connectivity matrix. And if you notice, we do a good job reproducing those connectivities, even though one comes directly from X-ray crystallography and the other one comes only by analyzing data, the sequence data. Okay, so this tool can be accessed through our lab website, morcoslab.org. But obviously, I'm happy to talk to someone who could be interested in using this. Now, I wanted to move a little bit faster past the DCA methodology, because I wanted to share a little bit more about this new tool that we're finishing. We're going to release very soon. And this is, again, a little bit more specific to people interested in signaling networks. So there's a signaling network called a two-component system. These are communication systems, mainly in prokaryotic systems and bacteria. And it's a way for the cells to internally communicate responses. Let's say chemotaxis or response to stress, even sometimes antibiotic resistance or circadian cycles. They use a protein as a sensor that then later transmit a signal to another protein called a response regulator, and then this response regulator in turn turns on let's say a response in the cell towards those inputs. Now, although these two-component systems are really abundant because they're basically in a big percentage of bacterial systems, they have only been really studied in a fraction of organisms, those model organisms that are typically studied, but these are really prevalent in thousands of organisms. Sometimes when someone is interested in a new organism, they might not have all the details because no one has really studied those in this new organism. Okay, so this is basically just a representation of what is the problem here. So in every single cell, you could have a multitude of these kinases on top. The molecules on top are kinases. These rectangles are representations of the membrane. So these are membrane bound. And then you have response regulators that are in the cell. Now, typically, this protein is phosphorylate, so they attach a phosphate group, and then they actually find its partner. They have a very specific encoding so that they only can recognize one or the other partners. So there are thousands of these in bacteria, and sometimes it's hard to identify which partner is the right one, right? So the way we did it is we use these methodologies of network inference to create a tool that will allow other people to look at organisms and then see if the preferences are confirmed with experiment, but they also can play a little bit with the networks and see what are the effects of mutations. So yeah, we call this evolutionary links infer for histone kinase sensors interacting with the response regulators, or ELIXIR. And it's gonna be useful to study from a network perspective how these interactions happen. This was developed by Dr. Claude Sinner. He's a postdoc in my lab and also a student, Yun Ho, who's been working on this. So what I want to show in the just last few minutes, I just wanted to show you a demo of this this tool and see if you find it useful. Yeah, this is going to be just brief, but I just wanted to give you an intro. So this is the website. There's a tutorial, and the main part of this web server is a list or a search of organisms, because we want really to analyze all possible interactions between these histone kinases and response regulators. For instance, if one searches an organism like E. coli, which is a very common model organism, then we can click there, and then this is the output of our algorithm, where we have a list of all the response regulators in E. coli and all the kinases that are found in E. coli, and then we have a metric of its specificity. So this bar shown at the right, so you have negative numbers for really specific partners, so pairs that are really specific, and red or positive numbers for things that are not supposed to be interacting. So you can go ahead and then, for instance, there's an example of NTRC, is a response regulator that is known biologically to interact with a partner called NTRB. So when we click here this link, then on the right-hand side, then we have a bar graph showing this preference. So this is very useful because someone who's interested in this particular protein might find what is its partner. If in this case, because it was known to be its partner, so it's evident, it's clear, but sometimes this is not known for other organisms, right? So you can export this bar graph so that you can use it in publications. But then you can also study other organisms like, let's say, other response regulators like Q7, and then you keep adding all this kind of measures of interaction. So you see that now this QSEV response regulator interacts with QSEC, it's another known interaction. Now, where things get really interesting is when we click on this particular interaction. So we click in this particular interaction, and then if we click mutate, then we can actually access the sequences, and then we can copy, doing some sort of in silico experiment where we copy parts of one protein, and then we try them in a different protein. Then when we modify, so we're doing a modification of the protein that accounts as a kind of a mutation experiment. So when we evaluate this, then we notice something really interesting, because now this change that we incur in cos R actually changes its partner, right? So we can see it here. So this is a change that would happen in theory, according to our prediction, if there was a mutation in that protein. So people can come and then test some of this hypotheses, then they can basically create their own mutations and then see if the connectivity matches their experiments, or if they want to propose a new experiment, they can test it here. This is something that is fairly new, has not been done before. People can also select specific interactions that they care, and then have them as a table that they can export. And they can also do an advanced search if they are, for instance, only interested in a specific search, right? And then they can display this particular search for only one protein with all its partners. So this could also be useful for publication or for specific analysis of residues. So we're adding more and more features, but so far we have 6,000 organisms, and our plan is to go and then keep adding more. This is possible to be applied to hundreds of thousands of organisms, but so far we are testing the first batch, first dataset. So I just wanted to end up with some acknowledgements for people in my group that work on this.
Thank you, Dr. Morcos. You do have questions in the queue. The first question is, are there any unique types of networks you've seen in biology?
I don't think there's, let's say, a unique type, so that there's only one particular type, but something that has been studied for a long time in terms of kind of network dynamics. I think this is the work of Laszlo Barabasi, was that in biology we tend to have what are called networks with nodes, so small world networks, I think they're called, or Justin probably knows the right terminology, but the idea is that you have elements that are considered nodes, like in the case there are master regulators that interact with many other nodes. So rather than having every node interacting with everyone else, we typically have a few nodes that interact with many others. So I think that's the unique kind of feature of biological networks, that they tend to be like small world networks. Yeah.
Excellent. Next question. What happens if the bacteria components cannot find their partner?
So if they cannot find their partner, so if I understand correctly, it means that if one single protein cannot be interacting with another, even though they are present in the cell, is that correct? Because in general, what we're assuming is that these components might interact. So in a cell, they're around, but sometimes we have information about their cellular localization, right? If we know that in advance, then we know that they will never get close to each other, so they might never interact. But the thing is, what happens is that because these molecules are built in such a way, in a very generic way, like with sequences that evolve, then even if we put them, once we put them together, if they have the right properties, they will interact. So sometimes the cell creates these divisions, this compartmentalization, so that they will not interact, so that there's no noise. but basically this is also, that's a good question, because it's also something that can be studied here. The idea of crosstalk. If I have a new protein that maybe interacts with others that is not supposed to interact, but if you put them in the right place, they might induce noise. Yeah.
Dr. Morcos, you do have another question in the queue. How different is this type of analysis to purely experimental methods? Could you give a perspective of time versus accuracy?
Yes. So it depends on which tool is the question referring, but let's talk about the first one. So it really depends a lot on the data that is available. So if you have really large amounts of data that is not very that is diverse, as we call it, then we can get as accurate almost as the X-ray. Now, the X-ray crystallography, it is really a gold standard. The thing is, doing X-ray for each protein, it takes time and it's expensive. So it's really a complementary thing. So if we have not enough data, then the quality might not be as good. So it really depends on the application. But we are encouraged by many other studies that have used this methodology to say something about not only structure but dynamics. Yeah.
Thank you, Dr. Morcos. Next up, we have Dr. Summers.
Dr. Tyler Summers is an assistant professor in the Department of Mechanical Engineering at the University of Texas at Dallas, where he directs the Control, Optimization, and Networks Laboratory. Prior to joining UT Dallas, he was an ETH postdoctoral fellow in the Automatic Control Laboratory at ETH Zurich from 2011 to 2015. He received a PhD degree in aerospace engineering with emphasis on feedback control theory at the University of Texas at Austin in 2010. He was a Fulbright postgraduate scholar at the Australian National University in Canberra, Australia, in 2007 and 2008. He is the recipient of a Computer and Information Science and Engineering Research Initiation Initiative from the National Science Foundation in 2016 and a Young Investigator Program Award from the Army Research Office in 2017. His research interests are in distributed feedback control, optimization, and learning in complex dynamical networks with applications to distributed robotic networks and electric power networks. Dr. Summers, welcome.
So thanks to Tiffany and everyone at the Office of Research for organizing this, and all the participants for being here today. So today I'm going to talk about a topic called system identification, and kind of keeping with the theme of the 101 data analysis, and because my understanding is that the audience is fairly broad, the talk will be at a fairly high level. But I'm happy to interact with anyone who's interested in more details, and I have some references at the end that give a lot more detail than what I can cover today. So here's the talk outline. I'll give a very high-level introduction to this topic of system identification, and give just a simple example that kind of illustrates some of the ideas. And then if there's time, I'll touch on a couple of more elaborate topics: subspace identification and sparse identification of nonlinear network dynamics, which connects to a couple of the previous presentation topics. So let's start with a kind of 30,000-foot view of system identification. So what is system identification? In one sentence, it's the theory and art of estimating mathematical models for dynamical systems based on observed data from inputs and outputs. So even more succinctly, you could say that system identification is data-driven mathematical modeling, but there are two keywords that come in this definition that really distinguish system identification from other types of statistical estimation or machine learning. And that's that we're talking about dynamical systems, so the behavior over time of systems, and also the fact that we have both inputs and outputs. And so these may be control inputs, things that we can manipulate the system with, things that as operators or designers of systems we can control, and then outputs that we can measure using sensors. So this is a very general mathematical framework, just to give you a flavor for the breadth of this. It has applications in engineering and biology and economics. So on the left here, some examples of inputs, and on the right, the corresponding outputs. So we might be interested in how, if we wiggle around the aileron of an aircraft, which is a control surface at the tailing edge of a wing, what effect does that have over time on the pitch rate of the aircraft? For a person who has diabetes, how do insulin injections from an artificial pancreas system affect over time the blood glucose levels as outputs of the system over time? In a power grid, how do generator power injections have an effect over time on the bus frequencies in the network? In an array, in a neuronal network, how do microelectrode array stimulation signals have an effect on the firing patterns of neurons in a neuronal network? And then a couple that we've been experiencing lately: how do social interventions like wearing masks or restricting certain businesses, how does that affect coronavirus infection rates? And how do economic stimuli like changing interest rates or other types of stimulus packages, how does that affect things like unemployment and economic growth over time? So what we'd like are mathematical models that allow us to describe relationships between these inputs and outputs, and we can use those models to make predictions and to do design and even control of these types of systems. So what are mathematical models for dynamical systems? What do they look like? So here's a very general representation of a mathematical model for a dynamical system. So first, let me just describe some of the signals involved here. So we'll collect all the inputs together and collect those in a vector called U. So those are the inputs at time t. We'll also collect in a vector all the outputs from all the sensors that are in the system that can make measurements for us, and we'll call that Y, so that's YT. And there's a couple other important variables. So there's another vector xt, which represents internal states of the system that typically we cannot directly measure, but that are important for describing the dynamics of the system. And then we have WT and VT. These are process and measurement noise that describe uncertainty or randomness in how the system behaves or how our sensors measure stuff about the state. So the model is described, a mathematical model is described by these two equations in this block here. So the first equation is a state dynamics equation. On the left-hand side is either the state derivative or a forward shift, time shift, the state at some time in the future. And that is modeled as some function of the current state, the current input, and then this random process noise. And this function's parameterized by a set of model parameters, which I've called theta. And then the second component of the model is the second equation, which is the output equation or the measurement model, which tells us how do the sensor measurements depend on the current state, the input, and this random measurement noise. And that's this function h, which is also parameterized by this vector of model parameters theta. Okay, and it's very important to keep in mind that this model, the mathematical model of the system, should be distinguished from the real physical system itself. So this is always just an abstraction. And there will be varying levels of uncertainty for different types of models and how well they can describe the input-output relationships in a dynamical system. So broadly, the way this works is that we collect some data, and again, since we're dealing with dynamical systems, we're going to get time series data, both of the inputs and the outputs of the system. And we'd like to do some processing on that data. So this, in general, we're going to just apply some statistical inference methods, and those will allow us to estimate the mathematical model. Specifically, we're going to get an estimate of the parameter that specifies these functions that I talked about in the last slide on the mathematical model. And we might also not just want a single estimate of this parameter, but we also might want to know something about the model uncertainty. So, for example, what's the probability distribution around this estimated parameter given the data that we've observed? So what we'd like for our model is not just that it fits well with the observed data. We really want this to generalize to data that we haven't seen before, so that it makes good predictions or allows us to make good designs of systems that operate on the model. We can view this as a type of machine learning, but again, the special thing about system identification is that the data are time series, and the models are these dynamical models that I described in the last page. Another way that this kind of distinguishes from other types of statistical estimation is that this gives an opportunity for experiment design. So again, some of the inputs are things that we might be able to control. We might be able to decide how to apply certain inputs to the system. And this leads to interesting questions about how can I design the input signal so that I make the best estimate of whatever model that I'm trying to estimate for the system. And then in the field that I'm in, which is in control systems and control theory, we might not just want to use the model for making predictions or for explaining some phenomena, but another common use of these types of models is that we can use them to design monitors and controllers that can control the system using feedback control. And so this actually becomes interesting because you don't necessarily want your model to be exactly capturing every single feature that describes the dynamics. You might be able to, you might be willing to give up some accuracy in your model if it simplifies your method for designing things like monitors and feedback controllers. So this is kind of just a broad workflow that summarizes some of the things that I've already said. So the way, and this kind of emphasizes that this procedure is an iterative procedure like many engineering design methods. So you start with an experiment that generates a set of time series data for the inputs and outputs in your system. And then you choose some sort of model class, so the set of models that you want to try to represent the relationship between the inputs and outputs. You feed that into a statistical inference procedure, and out comes an estimated model and maybe some quantification of the model uncertainty. You validate your model, maybe by doing predictions on unseen data or designing a controller. If the model's okay, performs okay in the validation, great, you use it. And if not, you can then iterate and maybe do a new experiment with different inputs or try a more sophisticated model class and repeat this procedure. So let me just go through a basic example now. So here's a simple model structure that's very commonly used in system identification. So this up here in the blue box, this is a linear dynamical system, so that the right-hand side of this equation here, xt plus 1 is ax plus bu plus the noise w. This is a linear function of the state in the input. And these parameters, which are the theta, are these matrices A and B. So if the state is an n-dimensional vector, A is going to be an n by n matrix. And if the input is an m-dimensional vector, the B matrix will be an n by m matrix. So these are some matrix parameters that we want to estimate. And then we might model this noise as being drawn according to some probability distribution that has zero mean, say a Gaussian distribution. And the idea with this basic model is that we're going to assume that we can measure the state vector and we can manipulate this input signal u. And one thing that we can do is just excite the system by generating the input signal randomly. So the random signals are actually a good way to excite all the dynamics that might be present in your system. So at every time t, you just draw your input signal from a normal distribution, say, that has a certain covariance matrix U, and then you do that over some finite time horizon and collect the state and input data. So you have these time series trajectories of the states and inputs, and we'd like to use that data to estimate this unknown parameter, these matrices A and B. And one simple way to do that in this case, since everything is linear here, is we just solve with this linear least squares problem. So we minimize the sum of the squares of kind of the equation error, the state at time t+1 minus these parameters times the state at time t and the input at time t. And you just solve that optimization problem. This least squares problem can actually be solved in closed form very efficiently. So the way this works, just more specifically, you can stack all of your state data into this vector x, and then stack the state and input data shifted back by one time unit into this matrix Z. And here is the general form for this model estimate that theta had. So it just involves some matrix multiplications and inversions to get this estimate. Okay. And once you have that, now you can use your model to make predictions on new data, or even design a feedback controller. So I can use these estimates of A and B to design a controller so that now I can have a feedback controller that changes my input based on the current measurement of the state. This is something that would be done often in the field of control theory. So like I mentioned before, we don't just want our data or our model to fit the given training data. We also want it to generalize. And one very common method that's used all over statistical estimation is this concept of regularization. So I want my model to fit the data, but I'm going to also add this penalty with a penalty parameter lambda. So this term R on the right here is called a regularizer. And you can think of this as a way to trade off how well the model fits to training data with how complex the model is. So the regularizer you can think of as like a penalty on the complexity of the model. And what this does, it can prevent your model from overfitting to your training data so that it promotes the generalization properties of your model. It also does something called striking a trade-off between bias and variance. So this is a well-known trade-off in statistical estimation theory. There are also Bayesian interpretations for regularization. So if you're familiar with Bayesian estimation theory, certain regularizers can be interpreted as imposing certain priors on the model. So some common regularizers are just the 2 norm of the model parameters or the 1 norm of the model parameters. So those are given down here. And I just want to connect with what Justin Ruths talked about earlier. The second regularizer, this 1 norm regularizer, has a nice property in that it promotes sparsity in the model. So suppose we had that model, this linear dynamical system, and we knew from some prior knowledge that we expected that there to be some underlying but unknown network structure in the model. In other words, we expect that the matrices A and B are going to be sparse in the sense that many of their entries are going to be 0 and a few of them are going to be non-zero that represent kind of these interconnections between the nodes in the network. And it turns out that this 1 norm regularizer term, so the theta, the 1 norm of the model parameters, if you add that into the inference procedure, it's known to promote sparsity in the model. And there are properties which I'm not going to talk about, but you can provably recover exactly unknown structure that's happening in the dynamics of the network. One cool thing is that this works on ill-posed problems. So if this is a big network and these matrices A and B may be large matrices with lots of parameters, you might be in a regime where the amount of data that you have is less than the number of parameters that you're trying to estimate. This is known as an ill-posed problem. But if you exploit this prior knowledge that you know that there should be a sparse structure underlying this, then even though you have less data than parameters that you're estimating, you can still provably recover the unknown structure. And then one last thing, with the 1 norm term, there's no analytical solution for the inference problem like there was for the least squares, but there's still lots of software packages that can efficiently solve the estimation problem with the one norm regularizer.
Thank you, Dr. Summers. The question that has been posed is, how can you use an identification system to analyze your prediction? And do you have an example of that?
So once you have the model, so for example, let me go back to just the simple example that I went over. Once you've identified these parameters, these matrices A and B, you can now do a simulation of the system that predicts what's going to happen to the state over time, given some input trajectory. So you can now, once you've done the model and trained it on some set of data, you can look at a new data set, or you can even do a new experiment with a different input and now see how your model, how the prediction of your model for what the state does matches up with data from a new experiment. And yeah, this is a very common thing. So there's lots of examples in, so in all the applications that I mentioned, this is a very common thing that you'll do to validate your model. You want to, again, you don't want it to just fit the training data, you want it to generalize to new data. And that's a common procedure that's done in all these application areas.
Thank you, Dr. Morcos, Ruths, and Summers, for sharing your time, talent, and resources with the attendees at this Data Analysis 101 workshop. For additional information related to Office of Research events, please visit research.utdallas.edu. Thank you.